Welcome to PhD in Science. Today's topic will be photosynthesis. Feel free to skip to the part that you're interested in and pause or rewind to the step that you didn't quite catch. If you don't understand something, feel free to comment below and I will be more than happy to answer. Without further ado, let's get started. Photosynthetic organisms such as plants, algae, or certain bacteria play a key role in sustaining life. Such photoautotrophs introduce chemical energy and fix carbon into ecosystems by using sun rays to synthesize sugars, such as glucose, for example. During this process, water is oxidized, so it loses electrons to form oxygen, while the carbon dioxide is reduced, so it gains electrons to form glucose in the plant cells. Both products have their use. While oxygen is released back into the atmosphere, glucose is stored inside the plant. Humans and other organisms, such as fungi, protists, prokaryotes, and animals that can convert carbon dioxide to organic compounds themselves, are termed heterotrophs. These individuals are obliged to get fixed carbon by eating other organisms or their byproducts. In most plants, photosynthesis usually takes place in the leaves. The cells in the middle layer of a leaf tissue are called the mesophyll, and they are the primary site of photosynthesis. There are also small pores, termed stomata, located on the surface of leaves. They are responsible for releasing oxygen and absorbing carbon dioxide. In mesophyll cells, there are chloroplasts, in which photosynthesis occurs. Furthermore, inside the chloroplast, there are disc-like structures called thylakoids. Their membrane contains green-colored pigments called chlorophylls that absorb the sun rays. Different chemical reactions occur in the different part of the chloroplast. Light-dependent reactions and light-independent reactions. Photosynthesis in the leaves of plants involves many steps, but it can be divided into two stages: the light-dependent reactions and the Calvin cycle, so the light-independent reactions. Light-dependent reactions occur in the thylakoid membrane and require a continuous supply of light energy. The light energy is absorbed by the chlorophylls, further converted into chemical energy through the formation of two compounds, ATP and NADPH, a reduced electron carrier. During light-dependent reactions, water molecules are converted to oxygen gas. The Calvin cycle, or light-independent reactions, occur in the stroma and don't necessitate light in order to take place. Instead, the Calvin cycle utilizes ATP and NADPH from light-dependent reactions to fix carbon dioxide and form three carbon sugars, glyceraldehyde 3-phosphate, and other molecules that bind to form glucose. Glyceraldehyde 3-phosphate is produced by carbon dioxide combining with ribulose 1,5-biphosphate, short RUBP. The reaction is catalyzed by an enzyme called the RUBP carboxylase oxygenase, better known as Rubisco. Not all forms of photosynthesis are created equal. There are different types of photosynthesis, including C3 photosynthesis and C4 photosynthesis. C3 photosynthesis is used by the majority of the plants, so for example, wheat plants and cotton, and is named after the three-carbon compound called 3-phosphoglyceric acid, short 3-PGA, that is used during the Calvin cycle. C4 photosynthesis, on the other hand, produces a 4-carbon intermediate compound named oxaloacetate, first converted to malate and then which splits into carbon dioxide and a 3-carbon compound during the Calvin cycle. A benefit of C4 photosynthesis is that by producing higher levels of carbon, it allows plants to thrive in environments without much light or water, meaning that plants are better adapted to hot and dry environments and continue to fix carbon even when their stomata are closed. Lastly, there's also the crassulacean acid metabolism, which takes place for plants that live in very hot and arid environments, such as cacti. As there is a risk of losing water through the stomata, When it opens during the day, these stomata open during the night, absorbing carbon dioxide. This carbon dioxide is then fixed into oxaloacetate and converted into malate. That was it. I hope you have learned something new and would appreciate if you can use these videos to support your learning path. I do also have an Instagram and TikTok account where I will post short, concise clips of each topic.